Okay, welcome back, folks. Moving to chapter 3 now, and we're going to do some review of some of the macromolecules of the cell. We're not going to get into all of them, just some review of what is possibly the most important thing. Let me fix my chair. There we go. Just to review some of the most important stuff, and then adding on some new information like we've been doing. So first of all, we'll start with polypeptides and look at proteins and protein structure. So let's get started with that. Just a quick word of caution before we get going with this. Like I say, this is a review of the most important principles. I'm not going to cover a lot of stuff, but to understand cell biology, you really have to understand the structures and the functions of macromolecules, because the structures that you see appearing in cells are assembled from the macromolecules, and so the functions that we see emerging from the assemblages of the macromolecules are completely dependent on the structures and functions of the underlying macromolecular structures. So that being said, let's get into polypeptides and proteins to start with. So should be familiar with the idea that amino acids get linked together in a stepwise fashion into a linear polymer called a polypeptide by dehydration, also known as condensation chemistry. As we take these two hydrogens here from one amino acid and we condense them with this oxygen from another amino acid, we produce a peptide bond and we release a water. So for every two amino acids you link together, you produce a water. And so here we're linking a glycine together, and here is its R group, it's a single hydrogen, and here is an alanine, here is its R group, a methyl group. We link those two together and we get the molecule glycylalanine, and there is the new peptide bond there, and we have this repeating N-CC-N-CC backbone. The end with the free amino group here, which is usually in a charged form given the physiological pH of the cytoplasm or most cellular compartments. The N-terminus is usually positively charged, so it's acted as a base and attracted a proton and become protonated, and so this is the N-terminus, and the C-terminus is acting as an acid and has lost a proton and is negatively charged at physiological pHs, and so there is directionality here in this short polypeptide. It's a dipeptide glycylalanine. So there's directionality. When we link together amino acids to make polypeptides, we make what's called a polypeptide. Obviously, the protein does not appear until we have secondary, tertiary, quaternary structures possibly forming, and we have native conformations with actual biological functions. So even though we talk about the process of protein synthesis, the linking together of amino acids by translation produces a polypeptide, which then undergoes a good amount of folding, which we'll talk about a little bit later. So thinking about secondary structures, you're familiar probably here with the alpha helix. Secondary structure refers to these local regions of localized folding that result from exclusively from the hydrogen bonding between the NH groups and the CO groups along the polypeptide backbone. So that's the NCCN. If you look at that backbone, you'll see NHs and COs. If you bring this close together, you get hydrogen bonding. There are two types of this, remember. There's the alpha helix, which you see here in the beta sheet, which we'll take a look at in a moment. Alpha helix, obviously spiral in shape, easy to mistake with the double helix, but there's only one strand, so should be able to keep that clear. The R groups project out from the alpha helix. There are about 3.6 amino acids per turn, and so this forms because a hydrogen bond will form between the NH group of one amino acid and the CO group of the next of the second amino acid, which is one turn away. So if you look at the amino acids here and you work out where the alpha carbons are, you can see where you have a hydrogen bond, and then look for one complete turn and then another hydrogen bond again, and that's what gives you this unique structure. So that's something a little bit new that you won't have heard about before. Beta sheets, these are kind of extended sheet-like conformations where the successive alpha carbons of the amino acids in the polypeptide backbone form kind of troughs like this or peaks like this relative to a plane of the sheet. So if the plane of the sheet is like this, and then we have an alpha carbon at the apex here, the next alpha carbon would be in the trough, and this gives us a repeating kind of sheet, like corrugated iron, if you've ever seen that on a roof. And then the R groups project from each side of the pleated sheet. Now, the unique thing about the beta pleated sheet is that they can be intramolecular. That means that we can have, like here, if this was all one polypeptide, then the hydrogen bonding is between regions of the same polypeptide. But you can also have beta sheets involved in bringing two protein faces together. So that's intermolecular hydrogen bonding between two beta sheets. That doesn't happen in alpha helix, so that's an important discrimination between alpha helix and beta sheet. Excuse me. Now there's two conformations of the beta sheet, and we'll take a look at these in a moment. They can either be said to be parallel, which is where the directionality in the two strands of some sheets is the same, and so this is where we would have C terminus to N terminus in one sheet, and then C terminus to N terminus in another sheet. So if they're running both in the same direction, they'd be parallel. If we have beta sheets and they're running in different directions, so N to C and then C to N, those would be anti-parallel. So they can have different orientations in that sense. So we have parallel and anti-parallel beta sheets. Give you a better kind of idea of these parallel, anti-parallel beta sheets, you can see here in these structural motifs, which we'll come back to in a moment. These structural motifs is what we have. Here are two beta sheets running in the same direction, and I know that because the arrowhead on the beta sheet here is always pointing towards the C terminus, and so this would be a parallel beta sheet, so would this one here, and then the one bottom left here would be an anti-parallel beta sheet. So these motifs are small kind of structural patterns that you find in a lot of proteins, and they're often made from different arrangements and combinations of beta sheets and alpha helix. Alpha helix and beta sheet are often going to be projected either as these sheets with these arrowheads like you see here, or as a coiled region for alpha helix, or a barrel. So the barrel here represents an alpha helix. And so there are these different motifs that you can see here. So this is a beta, alpha beta motif, and then a hairpin loop down here and a helix turn helix. The different parts of secondary structure are often connected by these disordered regions of polypeptide. So there's no such secondary structure in these random coil regions. The polypeptide backbone is just disorganized, but it's used to link two secondary structure elements together to produce these structural motifs, and then these motifs are then organized into domains, which we'll talk about a little bit later. So there's a little bit on motifs, and we'll come back to domains. When we think about proteins, this is folded proteins, they then can go from secondary structure to having tertiary structure where many secondary structure elements interact. So this is very, very dependent on the amino acid sequence, because don't forget, tertiary structure is dependent on interactions between the R groups. It's neither repetitive, tertiary structure is not repetitive, and it's very, very difficult to predict as well. Eddie. It's very difficult to predict. So despite many, many man hours being spent looking at Eddie, stop it. Being spent looking at algorithms to predict protein structure, it's impossible to do that right now. We can predict short motifs, but as soon as you get a protein over about 100 amino acids, it's very, very difficult to predict protein structure. Is what we get when we get tertiary structures. We get these hydrophobic interactions between R groups, hydrophilic interactions, ionic bonds between oppositely charged R groups, repulsions between similarly charged R groups, van der Waals interactions, hydrogen bonds, and then interactions between reduced sulfur atoms to form sulfhydryl groups. So all of those interactions give us tertiary structures, and when we get tertiary structure, that's often going to be the most stable folded form of the protein, called the native conformation. So like I started to mention here, we've got two kind of types of fully folded proteins in the native conformation: fibrous proteins and then globular proteins. So we'll look at globular proteins in a moment. When you see a fibrous protein, though, it's going to have extensive regions of secondary structure. So here we can see extensive regions of beta sheet, and then on the right you can see keratin. So keratin is a good example of a fibrous protein, fibrin from silk, elastin, collagen that you find in skin, ligaments, that kind of stuff. Those are all good examples of fibrous protein. So keratin here is purely alpha helix, and so you take alpha helix, and then when you start to coil two of those together, you end up with protofilament, and then you can assemble those protofilaments into an intermediate filament, which is a form of cytoskeletal element, and then you bundle those into cells in hair, and those cells, except in the follicle, aren't living, but you end up with this long, rigid, straight structural protein that you find in keratin. That's the main protein of hair. So those are fibrous proteins. Most proteins, however, are globular. These are folded into much more compact structures, and they have a huge number of tertiary structures. So for every individual protein you have with a unique function, it's going to have a unique structure. Now there may be regions of structural similarity when you look at conserved proteins or related proteins, proteins that have a lot of homology, that is they descend from a common ancestor. They can have a complete mixture of alpha helix, beta sheet, or they can be a mixture of both. They're usually composed of regions called domains, and these domains often have very specific functions. For example, a kinase domain could add a phosphate group to a lipid or another protein. And so we're probably looking at a domain over here on the right, these beta sheets and this alpha helix and this unstructured protein polypeptide, and maybe another domain over here. And each of those domains is going to have a specific function that contributes to the overall function of the protein. So maybe this domain on the right is involved in binding to a substrate, which the domain on the left is involved in phosphorylating. So this could be a protein kinase, for example. So here's a nice example of a protein with two domains. Here is a discrete, locally folded unit of tertiary structure, and here again is another locally folded tertiary structure, which is going to have a unique domain. And so we can see, if we counted these up, we'd see anywhere from 50 to 400 or so amino acids making up these discrete structures. So they can go from pretty small to pretty big. And you can see there's a combination of alpha helix and beta sheet packed together to give tertiary structure in both this domain 2 and domain 1. So when you find a protein which has got multiple functions, you're going to usually find multiple domains with each domain doing a specific function. And that's how globular proteins are assembled. That's usually in a modular fashion with these various domains like you see here. So like we mentioned, predicting tertiary structure is incredibly difficult. There are complex algorithms, but you usually have to determine the structure of a protein using experimental techniques, and we'll talk about one of those in a moment, X-ray crystallography. I've put a couple of URLs on this slide. You can't click on them obviously, but you can. They're short and easy to type in, and they're kind of fun. There are a couple of games which were developed to crowdsource people's attempt at folding complex structures, proteins and RNA molecules and that kind of thing. So you can have a play around with those if you like, give you a bit of an idea about self-assembly. So let's finish on revisiting the idea of self-assembly. Protein structures we've seen here is a very good example of self-assembly. Many proteins will fold spontaneously. They will just fold up into their native conformation. Some will require some assistance by molecular chaperones so they don't form unfavorable conformations, which may be more energetically favorable than the actual conformation that we want, the native conformation. So we'll wrap up there. Keep thinking about this idea of self-assembly and the fact that a lot of structures in the cell arise because of the inherent structural properties of smaller components that are used to build the bigger component, and the bigger component then has properties which emerge from the assembly of the smaller pieces. So we'll wrap it up there. When we come back, we'll talk about a technique to predict and determine protein structure, X-ray crystallography. I'll see you soon.